Okay, moving right along with these nice short lectures. We're already into the fourth lecture here for chapter 21 on nucleic acids and protein synthesis. We've covered the ideas so far of basic nucleotide structure and then primary structure, and now we're ready to move forward into nucleic acids. Okay, our previous lecture on the double helix was getting us ready for this new lecture focus, which is DNA replication. So hopefully you'll enjoy this one that really makes use of the concepts we've been dealing with up until now. DNA replication involves separation of the parent DNA into its complementary DNA strand. So I mentioned earlier that it's important that these hydrogen bonds are stable enough to hold these two complementary strands together most of the time, but not so strong that we can't break them when we need to. And we see here we certainly do need to if we're going to have cells divide and replicate the DNA instructions that we need for every cell. So pairing the bases in each strand with new bases to form complementary new strands. That's nice, elegant chemistry. And then if we zip them up with a polymerase, DNA polymerase or RNA polymerase, depending on what we're talking about. Here it's DNA replication, so we can produce two new DNA strands exactly duplicating the original DNA, and we can go and do that again and again and again, and it's very nice that nature has figured this out, and humans have figured out a lot of this as well. So we can start to do some really interesting things involving DNA replication. Okay, so I mentioned the polymerase is zipping these things up. Well, that's not free, right? Everything requires energy. So the way we do that is energy from the hydrolysis of these nucleoside triphosphates in order to form the phosphodiester bond to each new nucleotide on the complementary strand to build that new strand. So there is an energy cost, and we should recognize at least adenosine triphosphate's a familiar energy molecule for us. And so here we see that any of these nucleoside triphosphates, I should say, can form that phosphodiester bond to get us a new nucleotide on the complementary strand. It also releases a pyrophosphate, as we see, and that's how we go down from a triphosphate to a single monophosphate. So during DNA replication, we get an enzyme, helicase, that unwinds the parent DNA at several sections. So we're not doing this one at a time, we're doing it in several spots. The DNA polymerase catalyzes the formation of 5' to 3' ester bonds of the leading strand at each of those open DNA sections. And these are referred to as replication forks. The lagging strand, now growing in the 3' to 5' direction, is synthesized into shorter sections called Okazaki fragments, and the DNA ligase joins those Okazaki fragments to give a single 3', 5' strand. So there you have it, the direction of replication. Here we see that as a cartoon, which makes it maybe a little easier to understand than that unwieldy wording on the previous slide. So you see we're unwinding, we get that leading strand that forms very quickly, and then that lagging strand forms in segments that are then joined together later. We see down below the parent DNA, the replication forks that form as a result of this leading and lagging joining there of the unwinding of DNA, and then of course we have our small Okazaki fragments, or however that's said correctly, apologies there to whoever's name that is, in order to form our new DNA strands and double our DNA and allow for cells to divide and make two cells out of one cell. Okay, so before we close this short lecture on DNA replication, let's pause for a learning check. In this case, you're asked to match the following to their definitions. And so you have 1, 2, 3, and 4, and then you have A, B, C, and D down below. If you would pause the video here while you connect A, B, C, and D to their 1, 2, 3, and 4 counterparts, please start us back up once you're ready to check your work. Good luck. Okay, so hopefully you arrived at these same answers. So we start off with my favorite one there. The short segments formed by the lagging strand are the 4 Okazaki fragments, or however that's said. For B, the starting point for synthesis in unwound DNA sections is 3, the replication fork. C, the enzyme that unwinds the DNA double helix in the first place, is 1 helicase. And then D, the enzyme that catalyzes the formation of phosphodiester bonds of complementary bases, is 2 DNA polymerase. So hopefully if you didn't have those already selected, you understand now why those are the best answer choices. If you're still struggling, you're just not getting it, it's not making sense, please do reach out to me. It's, unfortunately there's not as much in my tool belt as with the organic chemistry, but I'm certainly happy to help give you some guidance and suggestions based on what's worked for me here. Otherwise, if things are going well, as I hope they are, we'll see each other again in the next lecture.